In today's topic, we will be studying transportation of gases, that is oxygen and carbon dioxide. In this topic, we can expect the questions from oxygen dissociation curve, which is very important for NEET, and respiratory pigment, and bicarbonate. So how the carbon dioxide is transported in the blood. So the highest amount of carbon dioxide is transported in our body is as bicarbonate. So these are the questions, these are the areas that we need to cover in gas transport. So if we look at what is transportation of gases, it is the transport of respiratory gases, oxygen and carbon dioxide from the alveoli to various organs and from the various organs to our alveoli. So at the alveoli, blood is receiving oxygen and giving out the carbon dioxide, and our tissues, the tissues are receiving the oxygen and giving out the carbon dioxide. So the transportation of gases, at the very first, if you look at the oxygen, it is the lungs in which there is a structural and functional unit called alveoli, which are millions in number, and in physical solution, that is blood plasma. So as we know, in blood plasma it is only about 3% of oxygen that is getting transported. 3% of oxygen that is getting transported, and 97% of oxygen is transported as oxyhemoglobin, and carbon dioxide transport, that is between the tissues and lungs, where as carbonic acid, that is H2CO3 minus, as carbaminohemoglobin HbCO2, and as bicarbonate, that is HCO3 minus. So these are the various forms that carbon dioxide is transported, of which bicarbonate is major amount that is transported. Next, coming with the very first, transportation of gases, that is in physical solution, the blood has 55% of plasma where it is containing water. So 98% of our plasma is water, and in the water readily carbon dioxide dissolves. So in the previous session we got to know that there is a difference between solubility of oxygen and carbon dioxide. So carbon dioxide is highly soluble than oxygen, about 20 to 25%. So oxygen though slowly dissolves in water, but still it dissolves in the water. So about 3% of oxygen is carried by plasma in the form of dissolved state. So dissolved oxygen is transported to tissue through plasma. And the next one is as hemoglobin. 97% of oxygen is transported by hemoglobin, red colored pigment that is present, and it contains iron. So if you look at the hemoglobin, we say alpha 1, alpha 2, that is 2 subunit, and beta 1 and beta 2 subunits. So totally 2 pairs of subunits are present in hemoglobin, which is a quaternary structure of protein. So this structure of hemoglobin we have studied in detail in the chapter biomolecules. So if we say alpha 1 unit alpha 1, alpha 2, beta 1, beta 2, together they can carry 4 molecules of oxygen or simply 8 atoms of oxygen. So each alpha unit or beta unit can contain 2 oxygen molecules, that is totally now 4 into 2, 8 oxygen atoms. So now next coming with the oxyhemoglobin. So there are some conditions where oxyhemoglobin can form. That is when the oxygen partial pressure of oxygen or concentration gradient of oxygen should be more, that is at the alveolar surface. So O2 binds with hemoglobin in a reversible manner to form oxyhemoglobin. This is called oxygenation. Hb has 4 units, so each hemoglobin molecule can carry 4 oxygen molecules or 8 atoms of oxygen. So at the very first, the hemoglobin molecule will not directly take in all the 8 atoms of oxygen. First it will take one oxygen molecule. Let me write for you. Let us say the Hb molecule. So one Hb molecule first takes one oxygen molecule to become HbO2. That is 25% saturation, and this HbO2 gets combined with another oxygen molecule for HbO2 O4, HbO4, which is 50% saturation, HbO4 plus O2, that comes for HbO6, that is 75% saturation, and HbO6 plus O2, that comes for HbO8, that is 100% saturation. So altogether the Hb molecule, this Hb molecule will not take up the 4 heme molecule can carry 8 atoms of oxygen or 4 molecules of oxygen. So for example, a taxi driver is there and he is not sure about the passenger. First he will take one passenger and he will start moving, and he is very much confident about the fuel, and he can carry some other passengers in his vehicle. In the same way here, and hemoglobin Hb2 will carry one molecule of oxygen first. So at the end it will have 8 atoms of hemoglobin. Then what are the conditions? So at alveolar, partial pressure of oxygen should be more and partial pressure of carbon dioxide should be less. And whenever we say partial pressure of carbon dioxide is more, we should also assume that pH is less, that is pH is less, temperature is less, and also the carbon dioxide is less. So in that condition only oxygen can enter into hemoglobin. The next part is at the tissues level. So at the tissue level again dissolution of oxygen will take place. So Hb is leaving the oxygen part by part, again by 25%, 50%, 75% and 100%. Hemoglobin will leave the oxygen. That is, what are the conditions for that? Okay. The same oxyhemoglobin will reach the tissues, and at the tissues the carbon dioxide concentration will be more. Carbon dioxide concentration will be more, and when carbon dioxide concentration is more, it is also seen that temperature will be more, pH will be more. When carbon dioxide is more, these two concentration will also be more. And this is the condition where we can see the dissociation of oxygen. Or the oxygen level should be less, carbon dioxide level should be more, and that is the condition where we can see the dissociation of oxygen. So the next one is as oxyhemoglobin, as we discussed here. So if we read out here, in alveolar there is high PO2, low CO2. Whenever there is less carbon dioxide, you should understand that there is lower H plus ion concentration and lower temperature. Lesser the H plus ion concentration, the pH will vary. The next one is the amount of oxyhemoglobin or oxygen carried by the oxygenated blood is for every 5 ml of O2, 100 ml of oxygenated blood is required. So, or in other words, 100 ml of oxygenated blood can carry 5 ml of oxygen to the tissues under normal physiological condition. And concept is the next concept is oxygen hemoglobin dissociation curve. In a normal condition, we see that there is a saturation of oxygen with the tissues is sigmoid curve. It is a sigmoid curve obtained when percentage saturation of Hb with O2 is plotted against the PO2. It is observed that the dissociation of oxygen mainly depends on the partial pressure of carbon dioxide. Whenever the partial pressure of carbon dioxide is more and is more, the affinity of oxygen decreases. So if it will shift to right side. When it shifts to right side, it is completely according to Bohr's effect. We see that it is decreased affinity between oxygen and hemoglobin. Decreased affinity between oxygen and hemoglobin. And if the same curve shifts to left side, we call it as Ardane's effect. And here we see the increased affinity between O2 and Hb. So that is one condition. So right and left we need to remember. So at right it is decreased affinity between O2 and Hb, and at left side it is increased affinity between the O2 and Hb. So this is with the dissociation curve. And next, coming with the transportation of carbon dioxide. In tissues, carbon dioxide is high due to catabolism, that is the metabolism of glucose, and PO2 is low. In lungs, CO2 is low and PO2 is high. This favors carbon dioxide transport from tissues to the lungs. It occurs in three ways: as carbonic acid, as carbaminohemoglobin, as bicarbonate. So carbonic acid, in the presence of carbonic anhydrase enzyme, the carbon dioxide dissolves in water of the plasma to form H2CO3 minus, that is carbonic acid. And the hemoglobin can also carry carbon dioxide as carbaminohemoglobin, and the salt which is formed in the plasma in the presence of carbonic anhydrase is bicarbonate again. So where there is a chloride shift that we are going to study now. So as carbonic acid, so 7% of carbon dioxide is dissolved in 7% of carbon dioxide dissolves in the water present in the plasma to form H2CO3 minus, that is carbonic acid. That is one way of carbon dioxide transportation. And this carbonic acid is formed in the presence of an enzyme called carbonic anhydrase. So with this enzyme, the reaction rate is more. Next, coming with the carbaminohemoglobin. So if we consider the hemoglobin, the second one is 20 to 25% as carbaminohemoglobin. So Hb present in the RBC, combines with the carbon dioxide to form carbaminohemoglobin. And you have to consider with carbon monoxide. What happens at the carbon monoxide? We all know carbon monoxide is a silent killer, which is odorless gas. When this forms, it is carboxyhemoglobin, which is highly toxic. Okay, so never confuse with carbaminohemoglobin and carboxyhemoglobin. Okay, affinity of Hb is more in the carbon monoxide than to carbon dioxide than to oxygen. Okay, than to carbon dioxide. So this is the affinity of Hb. So for carbon monoxide, it is more, followed by oxygen and followed by carbon dioxide. So carbaminohemoglobin is the hemoglobin, conjugation of hemoglobin with carbon dioxide. Then the last one is transport of gases transport of carbon dioxide as bicarbonate. Okay. So the transport of carbon dioxide as bicarbonate, around 70% of carbon dioxide is transported as bicarbonate salts. Is it recording?
Yeah, recording.
Fine. Okay. So as a transport of carbon dioxide as bicarbonate, it is taking place about 70%. So if we look at the tissues, so let us assume this is the cells present in the tissues, and the blood vessels are bringing the blood. Okay. So for my convenience, I am just drawing this as a bit of, I just draw the RBC here. Okay, RBC. And now carbon dioxide is more here and oxygen is more here. Okay, and carbon dioxide partial pressure is less and here oxygen is less. Now, as we all know, oxygen from higher concentration will diffuse to the interstitial fluid, from the extracellular fluid present here or interstitial fluid, and from there it will enter into the cell. And at the same time, when the carbon dioxide is more and partial pressure of carbon dioxide is less here, it has to move to the blood present in the capillary. This is the blood vessel, okay, and we consider this as an RBC, okay, and this is the plasma. So let us assume, now the carbon dioxide here, okay, carbon dioxide, I'll take one more image. So this is the ECF between the cells and the blood vessel. In the blood vessel, we see RBC, we see the plasma. So now oxygen from higher concentration is moving into here, and carbon dioxide from higher concentration is moving into here. Now we are going to study transport of only carbon dioxide. So let us assume the carbon dioxide first dissolves in the water present in the plasma, that is only 7%, into H2CO3 minus in the presence of carbonic anhydrous enzyme, that is only 7%. So remaining 20 to 25% of carbon dioxide, what will happen to it? That carbon dioxide combines with HB to form carbaminohemoglobin. Right? And next, 70% of the carbon dioxide dissolving in the water present in the erythrocyte, that is RBC. This is an RBC, right? So it will dissolve in the plasma present in the water present in the RBC to form H2CO3 minus. As soon as this H2CO3 minus is formed, okay, it splits into H plus and HCO3 minus. Okay, H plus and HCO3 minus. This H plus combines with hemoglobin to form H plus HB, which acts as a buffer. See, RBC is very delicate molecule. It should not get denatured. Higher pH will denature the protein, right? In the same way here, H plus ion, if it is increasing, then it may denature the RBC. So HB acts as an combines with H plus, and H plus HB will act as a buffering agent. Now this HCO3 minus, one negative ion is let freely inside the plasma of the RBC, cytoplasm of the RBC. Now it will shift to the plasma. When once it shifts, at the simultaneously what happens here? There is a salt, sodium chloride, which splits into sodium and chloride. This chloride ion, being the negative ion, will move into the cytoplasm of the RBC, which we call it as chloride shift. Chloride shift. What happens when? When HCO3 minus enters into the plasma of the blood? Here the chloride ion will move into the cytoplasm of the RBC. Shifting is taking place. Chloride is shifting from the plasma to the cytoplasm of the RBC, and we call it as chloride shift. So meanwhile, this HCO3 which is present inside was present inside the RBC, and now it is present inside the plasma, becomes Na plus HCO3 minus. This is called bicarbonate. Which bicarbonate? Sodium bicarbonate. Right? Sodium bicarbonate. This is the sodium which is the most efficient method. Also non-toxic method. Okay, here the blood is not getting any acidic since it is present conjugated with the sodium. Okay, sodium is an ion which combines with HCO3 and keeps the blood alkaline. Okay, so now this is at tissue level. At tissue level, what were the condition? Carbon dioxide were more. If we study in the previous session, it was 45 mm Hg. Carbon dioxide is more. Temperature is more. pH is low. That is H plus ion concentration is more, and the blood is acidic. In all these conditions, again oxygen is less. In all these conditions, what is happening? Carbon dioxide is entering from tissue to the blood in three ways. One is in the plasma. One is in the form of water present in the plasma. Carbon dioxide dissolves in the water in the presence of carbonic anhydrous to form what is this? H2CO3 carbonic acid. And 20 to 25% with hemoglobin to form carbaminohemoglobin. 70% in the water of the cytoplasm of the RBC to form H2CO3, again carbonic acid. When once this is formed, it splits into H plus and HCO3 minus. H plus combines with Hb to form a buffer agent, that is H plus Hb, and HCO3 will enter into the plasma of the blood where it combines with the sodium to form sodium bicarbonate. When once the negative ion has moved from the cytoplasm to plasma, one more negative ion should shift, that is chloride is shifting to the cytoplasm of the RBC, managing the electrolyte balance. So this is at the tissue level, and the same thing will repeat in the reverse order at the alveolar surface where so let us assume the alveoli. Okay, this is the thin squamous epithelium. This is the basement substance, and this is the capillary. What is this one? This is the capillary, and this, let us assume this is the same red blood cell which has in there, and we have plasma, carbon dioxide which was converted into H2CO3 minus by H2CO3 minus by the combination of carbon dioxide and water. It is reverse reaction will happen where now here carbon dioxide concentration is less because atmospheric pressure from there the alveoli is having lower carbon partial pressure. So carbon dioxide is more here, it will enter into the alveoli, that is 7%. And next 20 to 25% of carbaminohemoglobin that was formed here again shift back into Hb and carbon dioxide. This carbon dioxide is also entering into alveolar air. This is at alveolar surface. At alveolar surface, what is the condition, students? It is high PO2, less carbon dioxide, less temperature, higher H plus, that is less pH. Okay, more pH. Less pH. Okay. So in all this condition, what happened? Carbon dioxide is disassociating from Hb. Next, coming with the here. So sodium bicarbonate, which was formed there. Okay, which was formed there, again shifts back to sodium plus HCO3 minus. Why? Because CO2 is more, carbon dioxide is less. So this HCO3 minus will enter into cytoplasm of the RBC. Now this meantime H plus and HB will also split to H and HB, where this H plus ion combines with this HCO3 minus to form H2CO3 minus. In reversible reaction, again this is splitting back to H2O and carbon dioxide. The carbon dioxide is entering into alveoli. How much percentage this one? This is 70 to 75% of carbon dioxide. So the deoxygenated blood which was bought, through pulmonary arteriole. Which arteriole is this one, children? Which capillary is this? Pulmonary arteriole. So this pulmonary arteriole, this is the condition. So 20 to 25% of carbamino hemoglobin, which was having carbon dioxide in it, is splitting back because carbon dioxide concentration is less. So it has to thrown out. So carbon dioxide will enter into alveoli. Then next, or whatever the reaction happened in the previous chloride shift, it is happening in the reverse order. So we call it as reverse chloride shift. Reverse chloride shift. So what had happened here? The NaCl, the Cl which was present here, as now enters back to the thing and forms sodium chloride back. Okay, sodium chloride. Is that clear? So this is called reverse chloride shift. It is also called Hamburger's phenomenon. Hamburger's phenomenon. So let's just go with the PPT. Let's see what we were studying. First we will have the division. So in the present we can just see here. So 70% of carbon dioxide is transported by this method in the presence of an enzyme called carbonic anhydrase, which is slightly present in the plasma too. At tissue site, it facilitates the following reaction. Carbon dioxide, water in the presence of carbonic anhydrase form carbonic acid. Again, when the concentration of oxygen is more and carbon dioxide is less, the carbonic anhydrase will form HCO3 minus plus H plus from the same H2CO3. In alveoli, the above reaction proceeds in opposite direction, leading to the formation of carbon dioxide and water that we studied in detail now. So every 100 ml of deoxygenated blood delivers about 4 ml of carbon dioxide to the alveoli. This is the same reaction we studied. So this is at the tissue level, this is at the alveolar level. So that is one thing. Next, regulation of respiration.